1. Overview of histological staining sections of tissues, in order to give them the color needed for the structures to appear when viewed in the microscope. Biological tissues stain because most of them are transparent. The contrast of staining between the tissues is useful in pointing out the differences between certain types of cells, tissue structure, and other subcellular components such as cytoplasm, extracellular matrix, and nuclei. Histological stains can be broadly classified based upon the kinds of tissues or structures they stain and their chemical composition. Some of the stains are more specific, hence targeting special cell types, organelles, or other extracellular components. Others are more general by staining all cells and tissues alike in the same way. 2. Histological staining principles. The chemical and physical interactions that involve the tissue and the dye are the basis for the basic concept of histological staining. These may include basic and acidic staining. While some colors are basic, others are acidic. Basic dyes, cationic, combine with acidic components like nucleic acids, that is, DNA and RNA, whereas acidic dyes, anionic, combine with basic tissue components like cytoplasmic proteins. This differential binding increases contrast between different parts of a cell. Specific affinity to cellular structures. Depending on their charge, solubility, and molecular size, dyes may have affinity to certain cellular structure. For instance, the negatively charged phosphate group has affinity to hematoxylin, thus binding nucleic acids. Differential staining. It is applicable in a situation where various regions of the tissue segment are selectively stained using different stains. This feature enables the differentiation of tissues and cellular structures. Hydrophobic and hydrophilic interactions. Some stains adhere to hydrophilic materials such as proteins, while others interact with hydrophobic materials such as lipids. Third, representative histological distemper. There are many staining techniques used in histology. Each stain has specific characteristics and uses. A few of the most common ones are listed below: A, H and E stain, hematoxylin and eosin. H and E stain is the most used stain in histology and pathology labs. It produces a general impression of the tissue structure and is often used as routine examination. Hematoxylin. Hematoxylin is a basic dye which stains acidic cell parts, such as the nucleus housing DNA, blue or purple. It is applied to reveal the content of the nucleus in cells because it is attracted to chromatin and nucleic acids. Eosin. Eosin is an acid dye that gives pinkish or red colors to the cytoplasm along with other basic cell constituents. Such structures containing collagen, extracellular matrix, and cytoplasmic proteins are all eosinophilic. H and E stain is considered one of the most useful tools in discrimination of normal from abnormal tissue architecture. The contrast between the nucleus and cytoplasm is excellent by using this stain. B. Pass stain or periodic acid Schiff. The pass stain is also used to identify other carbohydrate-rich materials in tissues, namely glycogen and polysaccharides. Its application is based on oxidation properties of periodic acid which oxidize carbohydrate structures, creating aldehyde groups which react with the Schiff reagent to form magenta. Applications. Pass staining is often used to emphasize mucins, fungal components, and basement membranes. It is used in kidney biopsies for the identification of changes in the basement membrane in diseases related to the glomerulus, and in liver biopsies to detect diseases related to glycogen storage. Masson trichrome stain. There is a three-color staining method known as Masson's trichrome, which is used to differentiate among muscle, collagen, and epithelial tissues. Generally, three dyes are used. Hematoxylin, clearly purple stains nuclei. Biebrich scarlet or acid fuchsin, stains keratin red, cytoplasm, and muscle stains. Collagen is stained either blue or green by aniline blue or light green. Applications. Masson's trichrome is very helpful in the study of scarring, fibrosis, and connective tissue diseases. It is also useful in the study of heart, liver, and kidney diseases as it facilitates easier discrimination between the muscle fibers and collagen in histological sections. D. Silver staining discoloration. Silver staining is a technique that utilizes visualization of certain features of the cells and tissues, particularly those that are poorly absorbed with conventional stains. This method takes its basis from the conversion of silver nitrate to metallic silver, showing at its tissue seat as deposits that are either brown or black. Applications. Reticulum fibers, nerve fibers, and certain microbes such as spirochetes are often stained with silver staining. It is also used in pathology to detect amyloid and in neurohistology to study the structure of neurons except adiposi,